Traumatic spinal cord injury begins with a single devastating event, but its effects echo through a person's life forever. To date, over 500,000 people in North America are living with the long-term effects of a spinal cord injury. Some long-term symptoms of these injuries include full body paralysis, breathing difficulties, chronic pain, and incontinence. Why do these symptoms persist? And is there hope that we can change this outcome? After a traumatic injury, damage begins to build up in the spinal cord. The spread of this damage can be mitigated by treatments such as surgery and rehabilitation. However, in many cases, cell death continues for months after injury, eventually forming a cavity in the spinal cord. This cavity, known as the cystic cavity, is a hole in the spinal cord filled with cerebrospinal fluid and inflammatory molecules. Around this cavity forms a dense scar called the perilesional scar. Together, the cystic cavity and perilesional scar prevent the formation of new connections between the brain and extremities and present a major barrier to regeneration. In recent years, neural stem cells have been proposed as a solution to this problem. Neural stem cells are cells that have the capacity to self-renew and give rise to the main cell types of the nervous system. Neurons are responsible for transmitting electrical impulses throughout the body. Glial cells play a supportive role. Some produce myelin to insulate the neurons, while others help to clean up debris. The hope is that when transplanted, neural stem cells will differentiate and replace the cells lost to injury. However, when neural stem cells are transplanted into the site of injury, two major barriers stand in their way. The first is the physical barrier of the perilesional scar. This dense network of fibers and cells prevents many of the stem cells from integrating into the site of injury. The second is the toxic injury environment, which pushes the cells to become glial cells instead of neurons. Without neurons to transmit electrical signals and form the bridge across the gap, these glial cells add little to the repair of the injured area. The Failings Lab has addressed these concerns with a unique and promising solution involving engineered neural stem cells. To address the first barrier of the perilesional scar, a subset of stem cells is engineered to express a scar-busting enzyme called chondroitinase. Chondroitinase breaks down the main fibers of the perilesional scar. To address the second barrier of the toxic injury environment, another subset of stem cells is engineered to express a growth factor called GDNF. This growth factor pushes the cells towards becoming neurons rather than glial cells. When these cells are injected near the perilesional scar, they encourage the growth of new neurons. As the neurons mature and grow in number, they replenish the injured area. The hope is that coupled with targeted rehabilitation, these new neurons will form connections with existing circuitry and allow functional recovery of the spinal cord. Engineered neural stem cells are a promising solution to spinal cord injury. With this therapy, people with spinal cord injuries can expect to have less pain, improved respiratory function, better bowel and urinary control, improved sensation, and restoration of limb function. While this therapy is within our grasp, further funding is required to validate the safety and reproducibility of these stem cells for clinical trials. Together, we can harness the powers of regenerative medicine to create a brighter future for people living with spinal cord injuries.